Okay, so urinary system consists of these following organs: a pair of kidneys, a pair of ureters, urinary bladder, and urethra. What are the main functions of kidney? This is very frequently asked in your viva. In your viva table, also theory question. Okay, functions of kidney. Kidneys help in purification of blood by ultrafiltration and removal of metabolic waste products. Kidneys conserve the body fluid and electrolytes and maintain their balance. Kidneys also function as an endocrine organ by production of these three hormones. First one is erythropoetin. Erythropoetin helps in formation of red blood cells, and erythropoetin production depends on the blood oxygen levels. Next is renin. Renin converts angiotensinogen to angiotensin 1, and that helps in blood pressure regulation. Next is vitamin D3. The kidney enzyme 1-hydroxylase converts the 25-hydroxy vitamin D3 produced in liver to active 1,25-dihydroxy vitamin D3. Okay. These are the presenting parts of the kidney. Kidney is covered by a thick connective tissue sheath that is known as capsule. Okay. Just beneath the capsule, you will see cortex. Cortex have cortical tissues and medullary rays. Next is medulla. In medulla, you will see renal pyramids and renal columns of Bartini. And deep inside, there will be renal pelvis. Renal pelvis consists of major calyces and minor calyces. Okay. So, see here, this outer covering is the capsule of the kidney. Okay, just beneath the capsule, this part forms the cortex of the kidney, and these are the pyramids and renal columns of Bartini. All these are together forming the medulla of the kidney, and here is the renal pelvis. Renal pelvis consists of these three major calyces are present here, and they are dividing into this minor calyces. Okay, and this apex of the pyramid is projecting towards the minor calyces and forming renal papilla of the pyramid, and base of the pyramid is directed towards the cortex. Okay, renal cortex. And this area, this cavity is known as renal sinus. Within the renal sinus lies this renal pelvis. Okay, and this renal sinus also contains some adipose tissue, nerves, lymphatics, blood vessels like this. Okay, all those are entering into the renal sinus and then are going to be distributed in the cortex and medulla. And all these structures, this renal pelvis is coming out, blood vessels and lymphatics are entering into the kidney through this hilum of the kidney. So this part is known as hilum. This is a depressed area near the medial border of the kidney, and this lateral border of kidney is convex. Okay, understood? And outermost covering is known as the capsule of the kidney. So these are the gross structure of kidney. Each kidney is a bean-shaped organ covered by a capsule, and medial border of kidney has hilum through which the vessels and nerves enter the kidney. At hilum, the renal pelvis or space continues as the ureter. The space that occupies the vessels, nerves and renal pelvis at hilum of the kidney is known as renal sinus. Renal pelvis is divided into two major calyces. Those are upper and lower, and each major calyx is divided into a number of cup-shaped minor calyces that show conical projections of kidney called the papilla. In gross examination with naked eye, each kidney shows these following structures: outer reddish brown cortex and inner pale medulla. Cortex receives 90 to 95% of the kidney's blood supply, hence it is reddish brown in color. So this we have discussed. So we will discuss the individual parts in details. So first is capsule. Capsule of the kidney is composed of two layers. outer layer of fibroblast and collagen fibers, and inner layer of myofibroblast, and their clear function is not known. Myo means they have some muscle fibers, okay, so it helps in mainly contraction. Next is renal pyramids. They are the triangular projections of the renal medulla, and the apical portion of renal pyramid faces towards the renal pelvis and base towards the renal cortex. You have seen the pyramids, apex is directed towards the medulla and base towards the cortex of the kidney. Renal cortex shows striations. Light lines are there which are 400 to 500 in number. They are extensions of medulla into the cortex, and these are called the medullary rays of Ferrin. Medullary rays contain straight tubules of nephron and collecting ducts. Okay. Cortical labyrinths are the regions of cortex between the adjacent medullary rays. Renal columns of Bartin represents cortical tissue, and that is extended between the adjacent renal pyramids. Okay. Next we go to the papilla. Papilla is the apical portion of each pyramid, and papilla projects into the minor calyx. Area cribrosa. It is the perforated area of the tip of renal papilla due to the opening of the collecting ducts. Okay. Cribrosa means small openings are present here. Concept of kidney, lobe and lobule. This is also asked in your viva. You clear the concept about what is lobe of kidney and lobule of kidney. Okay. And this is also important for blood supply of kidney. Blood supply of kidney is very important, long question. You also have to practice a schematic diagram for that blood supply. Okay. So concept of kidney lobe. Each pyramid with the surrounding cortical tissue forms a lobe of the kidney, and each kidney contains about 8 to 18 lobes, that is multi-lobed kidney. So lobes, number of lobes will be equal to number of the pyramids. Lobulation is a prominent feature of fetal kidney, but in adult kidney lobes are indistinct. Okay. Next we go to concept of kidney lobule. Similar to the secretory unit of excretion glands, collecting ducts of medullary ray and the surrounding cortical tissue constitute the kidney lobule. Okay, that means each of the lobe of the kidney consists of multiple lobules. Okay. So lobule in the central part, there is collecting duct, and those nephrons which are draining into each of the collecting duct, all those with surrounding cortical tissue, they are forming one kidney lobule. Okay. So kidney is dividing into these components. First is glomerulus. Glomerulus is a capillary tuft, and it has some mesangium cells lying within the capillary tuft. Next there is nephron. Nephron are of these three types, and it consists of Bowman's capsule and renal tubules. Three types of nephrons are cortical or subcapsular nephron, juxtamedullary nephron, intermediate or mid-cortical nephron. Bowman's capsule consists of podocytes. They are forming the visceral layer, and squamous epithelium, they are forming the parietal layer. And in between these two layers, there is a space known as the Bowman's space. And renal tubule consists of these three parts: proximal convoluted tubule, loop of Henle, and distal convoluted tubules. Okay, and these are the three type of nephrons: cortical or subcapsular nephron lying at the cortex mainly, just beneath the capsule. Next is juxtamedullary nephron. Juxtamedullary means they are lying at the junction between cortex and medulla. Okay, and intermediate or mid-cortical, there some part is lying in cortex, some part is lying in medulla. Mainly loop of Henle is lying in the medulla. Okay. See here, this is a histological section of kidney in low magnification. Like this you will see under the microscope. See these rounded structures you will see, these are the glomerular tuft. The white space around the glomerular tuft, that is known as the Bowman's space. Okay, and these small rounded structures around this glomerulus, all these are sections of proximal convoluted tubule and distal convoluted tubule. How will you differentiate between proximal convoluted tubule and distal convoluted tubule? Proximal convoluted tubule is more pink in color and its lumen is narrowed, okay, because there is cuboidal cells and microvilli are present. But in distal convoluted tubule, here also cuboidal cells but lumen is larger in case of distal convoluted tubule. And maximum sections are of proximal convoluted tubule. And these linear tubules, these are sections of the loop of Henle and the collecting ducts. Okay, they are entering into the medulla, they are entering towards the medulla. So this part is one medullary ray, and around which this part around surrounding the cortical tissue, this is forming one kidney lobule, and this is whole of the pyramid. Some more medullary rays will be included. Whole of the part, this pyramid and this surrounding cortical tissue, all those are together forming the kidney lobe. Okay, so here we are seeing a part of the kidney lobe. Understood? See here the sections is showing. This is light microscopic view. This like this you will see under microscope. This part is cortex, this part is medulla, and generally in the cortex you will see the glomerular tuft. So these small structures are the glomeruli, and these small rounded structures, these are the tubules. Okay, proximal distal convoluted tubules, and within the medulla there are the collecting tubules and sections of multiple blood vessels. Okay, kidney is highly vascular, so you will see sections of multiple blood vessels here also. So here cortex have been enlarged view of cortex, glomerulus, Bowman's space. This is sections of proximal convoluted tubule. It is stained deep pink, okay, and lumen is narrow. And this is distal convoluted tubule, lumen is larger. And here is some collecting tubule has been shown. This section is elongated. Collecting tubule means sections will be elongated. And here this is a section through the medulla. You can see ascending limb and descending limb of loop of Henle. Lining epithelium is varying in different parts of the nephron. We will discuss. Okay, so these are sections of loop of Henle, and these are the collecting tubules, sections of the collecting tubules. Okay, understood? So here is proximal straight tubule, thick ascending limb. There is thick ascending limb of loop of Henle. This is section of collecting duct. Sections of capillaries are there. Thin segment. This is also a part of the loop of Henle. Okay. So these are the parts of the kidney, nephron mainly. Parts of the nephron. This is glomerular tuft. Surrounding the glomerular tuft, here is the Bowman's capsule and the Bowman's space. After that, this is the proximal convoluted tubule, and this line demarcates cortex from the medulla. This loop is known as the loop of Henle. It has a descending limb of loop of Henle, and here is the loop of Henle, ascending limb of loop of Henle. Ascending limb also has a narrower part and as a thick segment. Okay, narrow segment and a thick segment. After the loop of Henle, you will get the distal convoluted tubule, and distal convoluted tubule will going to drain into this collecting duct. Okay, understood? So one collecting duct is receiving multiple distal convoluted tubules. So kidney nephron, structure of nephron you have read in your 12th. Yes, okay, so we'll see the histological part here. So see the epithelial linings or different parts of the nephron. Lining epithelium very important for your MCQs and viva. Okay. So here you can see this is glomerular tuft. Capillary lining will be simple squamous. Then this is Bowman's space. So the area surrounding the capillary, that is visceral layer, and outside is the parietal layer. Okay, so Bowman's capsule, parietal layer is simple squamous epithelium, and visceral layer is lined by the podocytes. Podocytes are food processes and they help in absorption of different materials. Okay, and after that you will go to proximal convoluted tubule. It is lined by simple cuboidal epithelium with brass border and basal striation. These are the important features of proximal convoluted tubule because maximum absorption occurs here. 80% of absorption occurs in proximal convoluted tubule, and you have known brass border helps in absorption. Okay, brass border means they have microvilli. Okay, and after that there is loop of Henle. It has multiple parts and gradually the epithelium is changing. So proximal thick segment, it is lined by simple cuboidal epithelium with microvilli like the PCT, and main loop of Henle, this which part is lying within the medulla, it is lined by simple squamous epithelium. Then in distal thick segment, it is again lined by the simple cuboidal epithelium. Okay, after that we go to the distal convoluted tubule. This is again lined by simple cuboidal epithelium. But here the brass borders are not present. Okay, that's why also the lumen is larger. After that we go to the collecting tubule and collecting duct. Here you will see simple cuboidal to columnar epithelium. Every duct you see cuboidal to columnar epithelium. Okay. Microscopic structure of kidney is the nephron. Microscopically you will see mainly different parts of the nephron. So nephron is the structural and functional unit of the kidney, and each kidney contains approximately 2 million nephrons. Each nephron consists of renal corpuscle and long renal tubule. Renal tubule is made up of proximal convoluted tubule, loop of Henle, and distal convoluted tubule. And this distal convoluted tubule connects to the collecting tubule. Okay, all these parts you have seen. And see, all these tubules are surrounded by plexus of blood vessels. Okay, and the plexus of blood vessel around the loop of Henle, this part is known as the vasa recta. Okay, so here the entering artery is known as the afferent arteriole, and the artery coming out is known as the efferent arteriole. Diameter of afferent arteriole is larger than the diameter of the efferent arteriole, and due to this difference in the diameter, the filtration pressure is created. Okay. So what are the types of nephron? Based on the location of renal corpuscles, nephrons are classified as cortical or subcapsular nephron, juxtamedullary nephron, intermediate or mid-cortical nephrons. Cortical nephrons, the renal corpuscles lie in the outer cortex and they have short loops of Henle that extend only into the outer part of medulla. Okay. Juxtamedullary nephrons, their renal corpuscles lie at the junction of cortex and medulla, and they have long loops of Henle and ascending thin segments, and they help in maintenance of urine concentration. Next is intermediate or mid-cortical nephrons. Their renal corpuscles lie in the mid region of the cortex and they have average length of loop of Henle. Okay. See here, this is cortical nephron, only small part entering into the medulla. This is juxtamedullary nephron. Okay, most of the part is lying in the medulla, and they have a long loop of Henle. So they are the responsible nephron for concentration of the urine. Okay, juxtamedullary nephrons. All these parts we have discussed already, and these are the collecting ducts. Collecting ducts is going to drain into the renal papilla. Okay. So renal corpuscle or Malpighian corpuscle, it can come as a short note. Okay, Malpighian corpuscle. So, and also asked in your viva, what are the structures forming the Malpighian corpuscle? So renal corpuscle is a spherical structure and it consists of this following: glomerulus, which is a tuft of blood capillaries, and Bowman's capsule. Bowman's capsule, also known as renal capsule or glomerular capsule. It is a cup-like double-layered covering of the glomerulus, and there is urinary or Bowman's space. It is a space between the two layers of Bowman's capsule, also known as urinary space. Okay, so these are the structures together forming the Malpighian corpuscle. Okay, so first we will discuss these in details. Glomerulus, it is a spherical ball of tuft of blood capillaries. It receives blood through the afferent arteriole and drains into the efferent arteriole. The diameter of efferent arteriole is less than that of afferent arteriole, and there is a vascular pole and a urinary pole. Vascular pole is the site of entry-exit of efferent arteriole in the glomerulus is referred as vascular pole. And urinary pole, it is opposite to the vascular pole, and Bowman's capsule continues as proximal convoluted tubule, and this pole is called urinary pole. Okay. So here you can see the filtration apparatus of the kidney. That is also an important short note. Okay, filtration apparatus of kidney. So it consists of these three layers. This is capillary wall. We can see the red blood cell within the capillary. Capillary wall is lined by endothelial cells, which are simple squamous epithelium with flattened nuclei. After that, you will get the basement membrane. Okay, basement membrane, glomerular basement membrane or basal lamina. And after that, you will see the visceral layers of the Bowman's capsule that is formed by these podocytes. And podocytes has multiple food processes. Okay, and these food processes are interdigitating with each other. Okay, and through the spaces between this interdigitation, the materials pass from the within the capillary lumen up to the Bowman's space. Okay, so it is a fenestrated area. Fenestrated. Here capillary wall is also fenestrated for helping in passage of different materials from blood to the Bowman's space. Only except the blood cells, all of the material passes into the Bowman's space. And these are mesangial cells lying near the capillary. Okay. So filtration apparatus of kidney, filtration barrier or apparatus consists of these three components. You practice a schematic diagram like this for filtration apparatus of kidney. Okay, it comes as a short note, or also may be asked what are the three parts of filtration apparatus in viva. So first is endothelium of the glomerular capillaries. See, this is the endothelium lined by simple squamous epithelium. Endothelium of glomerular capillaries are fenestrated and that has large pores. These are the pore diameter 70 to 90 nanometer. Then there is glomerular basement membrane. It consists of fused lamina of endothelium and podocytes of the Bowman's capsule, and it forms the principal component of filtration barrier and prevents the filtration of plasma proteins. Okay. Next is visceral layer of Bowman's capsule or podocytes. The Bowman's capsule has two layers, outer parietal layer which is formed by squamous cells and inner visceral layer of podocytes. Podocytes have processes called the pedicles or food processes, and they extend towards the glomerular capillaries. Filtration slits, they are elongated spaces about 25 nanometer wide between the interdigitating food processes, and they are called filtration slits and helps in ultrafiltration. Okay, mainly large proteins and blood cells are not filtered. Next is filtration slit membrane or slit diaphragm. It spans the slits between the adjacent food process. Okay, glomerular filtration apparatus, it is a semi-permeable barrier and that has discontinuous endothelium and epithelium but continuous basement membrane. Okay, that is also a schematic diagram. This is the fenestrated endothelial lining. This is glomerular basement membrane. This is visceral layer of Bowman's capsule formed by podocytes. Okay, and food processes you can see, and these are mesangial cells and mesangial matrix. This is also a schematic diagram. Endothelial cells with pores. This is basement membrane and these are the podocytes with food process. Okay, 3 layers. Okay, and there is presence of filtration slit membrane between adjacent food process. So all these are slit membranes. Okay, in between the food process, the gap is the slit membrane. And this capillary is also showing the pores. See, like this the food process are lying. These processes are interdigitating with the nearby process of the podocytes. Okay, and this gap area, this gap area is forming the filtration slit. Okay, here we are showing the relationship of podocytes to a glomerular capillary. The red one is the capillary, and note that the entire surface of the capillary is covered by processes of podocytes, and the bare areas being shown only for sake of clarity. Oh, okay, so these areas will be also covered by the podocytes. See here, this is electron microscopic view. Okay, you can see this interdigitating nature of this food processes over the capillaries. Okay, understood? You can see clearly the pedicles or processes of the food processes of podocytes. There is primary process, secondary process, like this, and there are some debris, proteinaceous debris, and in between the processes there is filtration slits. Some cell bodies have also been seen, and there are some pedicles. So this is scanning electron micrograph of podocytes, visceral epithelium of glomerular Bowman's capsule surrounding the glomerular capillaries. And this is transmission electron microscopic view, okay, of podocyte and adjacent capillaries in the renal capsule. Here also you can see nucleus of podocytes. These are the food processes. In between the food processes, there will be the filtration slits. Okay. So this is primary process. After that, there will be secondary process like this. And this is lumen of the capillary. You can see the blood cells. This is also a lumen of capillary. So mesangium. Glomerular capillaries are held together by mesangial cells and their extracellular matrix. We have seen mesangial cells, and these mesangial cells along with associated extracellular matrix are called mesangium. Okay. Histology of kidney is important, short note. Histology of kidney can come. So you practice a schematic diagram. Okay. So mesangial cells work like pericytes of capillaries and are embedded in the basal lamina of the capillaries. There are lattice cells. These are the mesangial cells along the vascular pole, and these cells form part of the juxtaglomerular apparatus. Okay, juxtaglomerular apparatus, you have seen the name in your 12th. You have read the juxtaglomerular apparatus. That is also important short note. Okay. So what are the functions of mesangial cells? Phagocytosis of aggregated proteins forms the glomerular basement membrane. Cleaning of glomerular basement membrane supports the podocytes, secretion of interleukin 1 and platelet derived growth factor. Unlike other phagocytic cells, those are monocyte derived. Mesangial cells are derived from smooth muscle cell precursors. Okay, mesangial cells have angiotensin II receptors, and these cells helps in regulation of blood flow through the glomerulus. See here, this is the basement membrane, capillary, endothelium. Slits are there, and these are the podocytes with food processes lining the visceral layer of the Bowman's capsule. And these are the mesangial cells. Some of these mesangial cells, extra mesangial cells, here these are present. They are forming the lattice cells. Okay, and they will secrete the renin. The lattice cells are part of the juxtaglomerular apparatus. So schematic diagram showing the renal corpuscle. This part is renal corpuscle and juxtaglomerular complex. Here it is showing the juxtaglomerular complex formed by these juxtaglomerular cells of this afferent arteriole. This is part of distal convoluted tubule containing the macula densa cells, and these are extra juxtaglomerular mesangial cells. All these are together forming the juxtaglomerular apparatus. Okay. There are specialized cells in the short segment of distal convoluted tubule known as macula densa, and modified smooth muscle cells, juxtaglomerular cells in afferent arteriole, and there will be mesangial cells. So this is atlas picture of this cortex of kidney. You can see the glomerulus movement space, and here it is showing the juxtaglomerular apparatus, juxtaglomerular cells, macula densa cells. Okay, and this is sections of the collecting tubules. These are the sections of proximal convoluted tubule. You can see they are more pink, and this is section of distal convoluted tubule, larger lumen. Here microvilli are present, here microvilli are absent. Okay, and it is less pink in staining. And some sections of blood vessels, section of arteriole and venule you can see, along with the blood cells within their lumen. So this is enlarged view, glomerulus, movement space, DCT. Okay, this is macula densa cells have been shown here, and here squamous cells lining the parietal layer of Bowman's capsule. Juxtaglomerular cells are also being shown here, and these are the sections of proximal convoluted tubule, and this is section of distal convoluted tubule. Here the food processes you can see around the capillaries, food process of the podocytes, and lining epithelium of parietal layer is simple squamous. Okay, and this is afferent arteriole having the juxtaglomerular cells. These are the macula densa cells of distal convoluted tubule, and here are the mesangial cells forming the juxtaglomerular apparatus. And here is the proximal convoluted tubule, you can see. And see the difference in diameter. This is afferent, this is efferent. Efferent is narrower. Okay. So juxtaglomerular apparatus, it consists of these three cells. Macula densa cells are columnar cells of distal convoluted tubule. Juxtaglomerular cells are modified smooth muscle cells of afferent arteriole, and they secrete renin. Okay, so juxtaglomerular cells secrete renin. And lacis cells, they are modified mesangial cells, and their processes form the lace-like network. Okay. So this is the function of juxtaglomerular cells. Decrease in blood pressure, there will be decrease in sodium concentration in the blood. These two things will stimulate the juxtaglomerular cells, and they will secrete renin. Renin helps in conversion of angiotensinogen to angiotensin 1. Then this angiotensin 1 is converted to angiotensin 2 in the lung with the help of angiotensin converting enzyme. And angiotensin 2 helps in secretion of aldosterone from the adrenal gland, suprarenal gland, and increase absorption of sodium and water, and increase in blood volume and blood pressure. So it helps in correction of blood volume and blood pressure. This flow chart is also asked in your theory paper. Okay, how the renin and other angiotensin, all these are renin angiotensin system. So juxtaglomerular apparatus is a group of specialized cells near the vascular pole of renal corpuscle. It consists of juxtaglomerular cells, macula densa, and extraglomerular mesangial cells or lacis cells. Juxtaglomerular cells, they are modified smooth muscle cells of wall of afferent arteriole, and their function is secretion of renin. Next is macula densa cell. They are specialized cells of distal convoluted tubule, and that lie adjacent to the afferent and efferent arteriole. Macula densa cells are narrow, columnar, and crowded cells. Other cells of distal convoluted tubule are cuboidal. Okay. Nuclei of these cells are crowded and overlapping. Hence, these cells are called macula densa. Lacis cells or extraglomerular mesangial cells. Mesangial cells near the juxtaglomerular cells modify to form the lattice cells. And these cells have lace-like network forming processes, and these cells show receptors for angiotensin II and probably regulate the glomerular filtration. The lining. So this is mesangial cells in between the capillaries and glomerular capillaries, and here around the surrounding the capillaries will be the podocytes in the visceral layer. In the parietal layer, simple squamous epithelium. PCT lined by simple cuboidal epithelium with microvilli, brass border. Proximal straight tubule or thick descending limb of loop of Henle. Here also cuboidal cells with brass border. Then in the thin segment of loop of Henle, there will be simple squamous epithelium. After that, in the thick ascending limb of loop of Henle, there will be simple cuboidal epithelium, and in DCT also there will be simple cuboidal epithelium. In collecting tubules, there will be simple cuboidal to columnar epithelium in collecting tubules and collecting ducts. And this many of the collecting tubules, they are joined to form the collecting ducts, and collecting ducts are going to drain into the papilla. Okay, and these are the capillaries, section of capillaries lined by simple squamous epithelium. So all these epithelium you will see in the section of the kidney. Okay. Okay, so sections of kidney and nephron, the vascular plexus all have been shown here. This is thin segment of loop of Henle, thick segment of loop of Henle, section of capillary. This is one papillary duct or collecting duct have been shown. This is distal convoluted tubule, proximal convoluted tubule. So renal tubule. Renal tubule consists of proximal convoluted tubule, proximal straight tubule, loop of Henle, distal straight tubule. All three are part of loop of Henle, and then distal convoluted tubule. The renal tubule receives glomerular filtrate from Bowman's capsule at proximal convoluted tubule and continues at distal convoluted tubule with the collecting ducts. Renal tubule is lined with simple or single layered epithelium. Proximal convoluted tubule is 40 to 60 micrometer in diameter. It receives the ultrafiltrate from urinary pole of the Bowman's capsule. It is lined by simple cuboidal epithelium with brass border, central spherical nucleus and pink eosinophilic cytoplasm. Electron microscopy shows the features of cells of proximal convoluted tubular as follows. There will be brass border. These are long, closely packed, straight microvilli. There are tight junctions. They are present on the apical portion of the cells. And basal striations, they are longitudinally oriented mitochondria, keep basal striated appearance. Note, brass border and basal striation increase the surface area of the cells for absorption. Function is reabsorption of 80% of ultrafiltrate, that is about 150 liter per day. Okay, 150 liter per day absorption occurs in proximal convoluted tubule. In PCT, sodium potassium ATPase pump and aquaporin proteins are mainly responsible for reabsorption of the water, and PCT also reabsorbs amino acids, glucose and polypeptides. Okay, so proximal straight tubule, it is thick descending limb of loop of Henle. It is communication between PCT and loop of Henle. Some authors consider proximal straight tubule as a part of PCT and some as thick descending limb of loop of Henle. The cells of proximal straight tubule are shorter than that of PCT, and these cells have less developed brass border, basal and lateral folding, and less mitochondria than the PCT. Okay, that is the difference from PCT. Next is thin segment of loop of Henle or ansa nephroni. The length of this thin segment of loop of Henle is variable according to the type of nephron. It is longest in juxtamedullary nephron, whereas shorter in the cortical nephrons. And in HE staining, the thin segment of loop of Henle is lined by simple squamous epithelium, even low cuboidal epithelium with few microvilli. There is pale staining cytoplasm, bulging elongated nucleus, and it resembles venule in the cross section. Okay. So this is the cortex. These are the medullary rays. medullary rays and surrounding cortex forming the kidney lobule. And this is whole of the pyramid. Pyramid with surrounding cortex is forming the kidney lobe. Here also we are seeing one kidney lobe. These are the glomeruli. Here are the sections of PCT, DCT. Here are sections of loop of Henle and collecting ducts. Okay, and this is renal papilla. Renal papilla is here. Here the collecting ducts are opening into the minor calyx. And there are some adipose tissue surrounding the kidney tissues. Here you can see kidney medulla, okay, papillary region, transverse section. You can see these are the sections of papillary ducts. These are straight ascending segment of distal tubule. Okay, here are straight ascending segments of distal tubule again, and this is straight descending segment of proximal tubule. There are some connective tissue sections of capillaries. All these you will see in the sections of medulla of the kidney. So mainly sections of loop of Henle, sections of collecting ducts. The schematic presentation. Medullary ray showing sections of collecting ducts and loop of Henle. Here PCT DCT section and glomerulus in the cortex. This is the enlarged view. Mesengium, glomerular capillary, Bowman's capsule, Bowman's space, PCT, sections of PCT, sections of DCT. Okay, the glomerulus, Bowman's space or urinary space. Here sections of PCT and DCT you can see. 4 is PCT, here is the DCT. Here juxtaglomerular cells, macula densa in DCT and juxtaglomerular cells in afferent arteriole has been shown. These are the DCT sections, some are PCT sections. Collecting ducts, elongated sections are of collecting duct, okay, and different renal tubules, PCT, DCT, glomerulus, parietal layer of Bowman's capsule. So distal okay, here this is section through the medulla. These are sections of loop of Henle, thin limb, and loop of Henle, thick limb is this section of loop of Henle, thick limb. And this is the 1A as seen in transverse section, and these are the longitudinal sections of the collecting duct. So Henle, loop of Henle and collecting duct sections you will see in medulla. Here also this is loop of Henle thin segment, loop of Henle thick segment, the sections of collecting duct, and some sections of arteries you will see. So renal tubule, distal straight tubule or thick ascending limb, part of ascending limb of loop of Henle, lined by large cuboidal cells with light pink or eosinophilic cytoplasm and apically located nuclei. Next is distal convoluted tubule. They are about 1/3 long as that of PCT, and DCT are lined by simple cuboidal epithelium with centrally placed spherical nucleus without brass border. These are the function. Its main function is reabsorption of sodium, secretion of potassium, reabsorption of bicarbonate ions that makes urine acidic, and DCT works under control of aldosterone for increased absorption of sodium ions. Okay. So these are the difference between PCT and DCT. Length, PCT longer, DCT shorter, 1/3 of PCT. Diameter is bigger, 60 micron, and DCT diameter smaller, 15 to 30 micron. Epithelium is simple cuboidal or low columnar, here simple cuboidal. HE staining of cytoplasm darker pink than DCT, light pink, light staining pink. Nuclei in each cross section is fewer, more and equally spaced. Lumen of tubule is star-shaped irregular. Here lumen is circular or oval and regular. Brass border is present, brass border is absent. Basal striation due to mitochondria is present in PCT, absent in DCT. Okay, so this difference is sometime asked in your viva or can be given as a short question in theory. Collecting tubules and collecting ducts. Collecting tubules are 40 to 50 micron in diameter. begin in the cortex as continuation of DCT and proceed to the medullary rays. In the medullary ray, collecting tubules join to form the collecting duct, and these collecting ducts run toward the apex of the pyramid. They join to form the papillary duct of Bellini, and duct of Bellini opens on the free surface of the papilla. In HE staining, circular cross section of collecting tubules and ducts show clear outline of the cells, and this distinguishes them from convoluted tubules. Those have irregular sections and indistinct cell outlines. Collecting tubules are lined by simple cuboidal epithelium without brass border, but it becomes columnar as the size of duct increases. Cell types are collecting tubules and ducts are lined by light clear cells and dark cells. These are intercalated cells. Light clear cells, these are called collecting duct cells. That is, these cells have single cilium and have short microvilli. These cells have ADH regulated water reabsorption aquaporin-protein 2 channels, aquaporin-2 channels, and defective aquaporin-2 genes causes diabetes insipidus. Okay, and these cells show basal infoldings. So dark cells or intercalated cells, they are few in number. These cells do not show basal infoldings. See here, this is cell of PCT, microvilli, tight junctions, mitochondria, more in number, and this is cells of DCT. Few microvilli is present, number of mitochondria is less, it is less cuboidal. Okay, and this is the basal nuclei, basal infoldings are present, basal lamina. Difference between cells of PCT and DCT. Main difference is this microvilli. Okay, here also difference between cell lining of PCT and DCT. More number of regularly arranged microvilli forming brass border. Lateral infoldings, mitochondria, nuclei, basal infolding. All these are also present here. Next we go to blood supply of kidney. Okay, renal artery divides into interlobar artery and vein. They are passing through the renal column of Bartini, and just above the renal pyramid they are bifurcating to divide into two arcuate arteries. Okay, from each arcuate artery interlobular artery is arising, and from the interlobular arteries they are giving afferent arteriole. Okay, afferent artery is going to form the glomerular plexus, then it will go to efferent arteriole, and efferent arteriole is form a plexus around the renal tubules, then they are joined to form this they will join this vein, interlobular vein. Okay, interlobular vein joined to form this arcuate vein. Arcuate veins are joining to form the interlobar vein, and these interlobar veins will join to form the renal veins. Okay, so that's how blood is circulated through the kidney. Understood? And there are some loops in the medulla. They are forming the vasa recta. And these are the plexus, medullary capillary plexus, here glomerular capillary plexus, there is peritubular cortical capillary plexus, there is stellate veins are present just beneath the capsule. Like this, there are multiple capillary formation within cortex and medulla. So renal microvasculature, each kidney is supplied by renal artery. Renal artery divides to form lobar and then interlobar arteries. Interlobar artery is branch and continue as arcuate artery along the base of the pyramids. Bifurcates into two arcuate arteries. Arcuate arteries give interlobular arteries, and that traverses cortex towards the capsule. Interlobular arteries give afferent arterioles. Afferent arterioles give rise to network of glomerular capillaries that unite to form the efferent arteriole. Efferent arteriole give rise to second set of capillaries known peritubular capillaries. Efferent arterioles from the juxtamedullary glomeruli form the vasa recta, help in countercurrent exchange system. And peritubular capillaries in cortex drain into interlobular vein and later into arcuate vein, interlobar vein, finally renal veins. and capsule drains into stellate veins and finally into the interlobar veins. So this is the blood circulation through the kidney. We have seen the previous picture. Previous picture you practice as a schematic diagram of blood supply. Blood supply is very very important for kidney. It comes as a long question in theory. Okay, blood supply. Also, if kidney viscera is given, you may be asked the blood supply. Okay, understood? See here, some this section of kidney renal artery is dividing into lobar arteries. Lobar arteries are again dividing into interlobar arteries, and interlobar arteries are bifurcates over the base of the pyramid as arcuate arteries. Okay, this is the glomerular plexus have been shown here. So this is interlobar artery dividing into two arcuate artery. From the arcuate artery, interlobular artery are arising. Interlobular artery from here as afferent arterioles are arising. Okay. And here are the capillary plexus, ascending vasa recta, capillary plexus around loop of Henle and round collecting duct. They are helping in counter current exchange phenomenon. Okay. Here interlobular veins are joining the arcuate veins, then join to form the interlobar veins, then lobar vein, renal vein. Okay. So here we see the capillary plexus around the convoluted tubules. There are the glomerular capillaries, superficial glomerulus, deep glomerulus. Okay, so arcuate artery, those interlobular artery. These are the nephron have been shown, collecting ducts, pyramids, afferent arteriole. This is cortical lobule, around the medullary ray. This is one schematic diagram. Okay, these are the glomeruli. These are the medullary rays, pyramid, arcuate artery, interlobular artery, interlobar artery. Okay, this is also showing the blood supply. So you can see the division of arteries. So this is showing the relationship of podocyte to the glomerular capillaries. It is surrounding the glomerular capillary. These are the food processes. Some mesangial cells are present in between the capillaries. This is vasa recta around the loop of Henle of juxtamedullary nephrons. This is peritubular capillary plexus, glomerular capillaries. So this flow chart you remember. Okay, renal artery, which is a lateral branch of abdominal aorta, divide into lobar artery, from there interlobar artery, from there arcuate artery, then interlobular artery, afferent arteriole, glomerular capillaries, efferent arteriole, in the cortex vasa recta, peritubular capillary plexus, in the medulla vasa recta, and this peritubular plexus join to form the interlobular veins going to drain into arcuate veins. In medulla, vasa rectas are going to drain in arcuate veins. From here, interlobar veins, they join to form renal veins. Renal vein is going to drain into inferior vena cava. And stellate veins from the capsule, they are draining into interlobular veins. Okay, so this is the blood supply. This flow chart you remember and practice a schematic diagram. So clinical aspects, glomerulonephritis or Bright's disease. It is the term used for diseases that primarily involve the renal glomeruli. It is convenient to classify glomerular disease in two broad groups: primary glomerulonephritis, secondary glomerular disease. Primary glomerulonephritis, in which the glomeruli are the prominent site of involvement, and secondary glomerular disease, it includes certain systemic and hereditary diseases which secondarily affect the glomeruli. Nephrotic syndrome, it is a constellation of features in different diseases having varying pathogenesis. It is characterized by findings of massive proteinuria, hypoalbuminemia, less albumin in blood, edema, hyperlipidemia, lipiduria, and hypercoagulability. So these are the features of nephrotic syndrome. Next, there is hydronephrosis. It is the term used for dilatation of renal pelvis and calyces due to partial or intermittent obstruction of the outflow of urine. Hydronephrosis develops if one or both the pelviureteric sphincters are incompetent, as otherwise there will be dilatation and hypertrophy of the urinary bladder but no hydronephrosis. Hydroureter nearly always accompanies hydronephrosis, and hydronephrosis may be unilateral or bilateral. Okay, hydronephrosis means dilatation of the pelvic calyceal system. Hydroureter means dilatation of the ureter. Okay, so these 2 points you have to write down for kidney slide in your spotting, and also write down in your histology copy. Outer cortex containing renal corpuscles and sections of proximal distal convoluted tubules are seen. Inner medulla, sections of collecting ducts and loop of Henle are seen. That's how you identify kidney slide. And this diagram you draw in your histocopy. Okay, this is glomerular capillary plexus movement space. This is the parietal layer lined by simple squamous epithelium. Here is sections of proximal convoluted tubule. Okay, and this is section of distal convoluted tubule. These are the sections of collecting tubules, and here are some sections of blood vessels. Okay, so all these sections you will see in a slide of kidney. Understood? And sections of PCT, DCT, and some sections of the capillaries you will see. Okay. And towards the right side, you will see some longitudinal sections of tubules. That will be the sections of the collecting ducts, okay, and loop of Henle. So that is the junction between the cortex and medulla. Okay, so on the left side you will see the cortex, outermost is the capsule. After that, cortical tissue consists of the glomerular mesangial corpuscles, glomerular capillaries, movement space, and some sections of PCT, DCT capillaries. Okay, that you see here. Okay, here also you see left side there is the cortical tissue, you can see some glomeruli, and on the right side you can see longitudinal section. Most of the part here it is showing the medulla, longitudinal sections through the collecting ducts and loop of Henle's and some capillaries. Okay, so here you see the medulla, how it looks, medulla and sections of PCT and DCT. Okay, and some in the middle there is a section of blood vessels, mainly arteriole. Okay, larger artery is seen in the middle. PCT, DCT you can identify from those different features, but generally we are seeing in our class in low microscope. It is not always possible to differentiate in low microscopic view. Okay, you just know there are sections of PCT and DCT. Here also you see the cortex, and in between you can see the medullary rays. Okay, the glomerulars are lying in lines. In between the glomerulars there are some elongated things. Those are the medullary rays formed by the collecting tubules. Okay, so the area of cortical tissue around the medullary rays together forming the kidney lobule, and whole of the pyramid along with the cortical tissue will form the kidney lobe. Okay, so you see the kidney lobule and cortex here. Cortex is clearly visible. So on these two slides you see cortex and medulla. Here is enlarged view of the cortex, and here also cortex and some medullary rays are clearly seen here.